Hello and good evening all. We welcome you to this online session on bioinformatics, and I hope people who have attended yesterday's session, that was an introductory session. We have told you certain rules about the program, what we will be doing, and why we have started this program, and what are the things that need to be taken care while we are doing this particular program. Now, as we have given a time of 5 minutes so that people can tell us or join us, because that is the time which we have given. So 5 minutes time we will give for the people to join, and after that joining over here is not allowed, because then if they want, then they can join on our YouTube channel and they can view it. However, as we have already discussed in yesterday's session and in the welcome email also which we have sent, that the certificate for the session will be given only when you will provide the feedback, and the feedback the registration, or I should say the feedback link, will be shared over here in chat. So the first thing which I would like to know is that are you all able to hear me? You can type it in the chat and just let me know that if you can hear me. Okay, yes. Thank you very much. So yes, people can hear me, so that's good. So what I will do is that we will wait for another 3 minutes. Now, in these 3 minutes, I will talk to you about this program which which we have started, and this program is the initiative of the BDG Lifesciences, which has come under the banner of Envision with BDG. So as you know that it is hard times for all of us, and we are really stuck up at our places, but then learning has not stopped, and then we've adapted to this situation, and we are doing online sessions, online trainings, and online what I should say, online workshops. However, I would like to tell you that BDG Life Sciences, previously it was called Biodiscovery Group, and we are a 10-year-old organization. Now, we are into online trainings and workshops and research projects since 2012. So this is one thing. So online mode is not new to us. Online mode is, we have been doing since long time. And as specifically I have told you, since 2012. So this is what we have been doing. And when this COVID-19 pandemic started, we have increased our the frequency of our online events more, so that a lot of people can join. Then we have started, as these are hard times, and it is already pitching, the picking up the pockets. So we have launched free courses, and I would like, I would proudly tell you that till now we have done more than 70 free courses, in which more than 14,000 people from all over the world have participated. Now, these people, they were not only students who were, bachelors, they were PhD students, research scholars, doctors, as well as company professionals and scientists. So this is what we have been doing since this 6 months or 7 months, the past 8 months of the COVID-19 pandemic. So I would like to take only a very few time to tell you about what BDG Life Sciences is, and then we can proceed from there, because our my colleague, Miss Sharvari Kulkarni, she M.Tech, she has already joined, and she is basically the trainer for our 7-day technical hands-on bioinformatics workshop, which we have done a number of times. And again, we are doing it from 12th of October. So you can check out the website for that. However, we will discuss later. Now, people who don't know about us, we are an international company which was started in 2009 in the form of group. And just keep in mind that we have started this company with a motive of help to those who really do not know after their bachelor's and master's that what we should do so to have much better opportunities for jobs and higher studies. So this was the reason why this company started. Let me begin with this particular slide. Now, this particular slide, if you can see, then it says that this is a quote by Benjamin Franklin, and it says that an investment in knowledge pays the best interest. Now, we all know that we have grown up, our parents have invested in us, and they've invested in our studies so that we can become good human beings, and that is the reason why they have invested in us and invested in education so we can become better human beings. And that is all. They've invested in knowledge so that it will pay a better interest in the form that we are now in a position where we can help others and we can have, we can earn our living in a much better way. What BDG Life Sciences is? BDG Life Sciences, it is a guide who will help you in solving your academic problems, showing you the right path, solving your queries, giving you correct training, paving your path for a successful career, and giving you launch pad to excel in the field of science. Now, I would like to elaborate on these points, which are very important for a student, especially the bachelor and the master's one. Showing you the right path. Showing you the right path means that we will guide you, we will help you in telling you or giving you counseling that what should be your next step academically, what should you do, which courses you can take up, which trainings you can take up, so that you can have a much better position to pitch in for your higher studies or for employment. Then solving your queries. Now, solving your queries is something really important, especially for the PhD scholars. Normally, PhD scholars, they stuck up at one point, every PhD student, they come to, there comes a point where they got stuck up and then they do not know where to go and how to proceed. So that is the reason they contact us, and then we solve their problems. Giving you correct training. Now, giving you correct training is something really important, because the education is that if we will teach you something wrong, then it will definitely destroy your knowledge. So giving the correct training is really important, because then it will help you. So that is the way how it should be. Paving your path for a successful career. Now, paving your path for a successful career means that definitely we help you in guiding you what steps are to be taken for your future, so that you can have a much better life and you can have a much better opportunity in employment as well as higher studies. Last point is giving you launchpad to excel in the field of science. This point is really important because we use our network to help you placed for jobs, for PhD, opportunities and even the postdoc positions. So basically, BDG Life Sciences, it itself tells you that we are a group who is working towards the goal, and the goal is to help you to make your career better, or I should say to give you an opportunity so that you can learn and excel into the field of biotechnology as a whole. However, we are a bioinformatics company, so it's our focus is completely on bioinformatics. However, this is the motive of BDG Life Sciences. So this is, you just keep in mind that we have started this organization in the form or with the motive. Okay, so the next thing which I would like to tell you is how we started. In 2009, the motive of starting the organization was in the form of group. Now, in the form of group is, till now you have BDG. Now, I don't know whether you know about BDG, what is the full form of BDG, but the full form of BDG is Biodiscovery Group. The name of the organization itself is Biodiscovery, but then we have added group. Previously, it was Biodiscovery Group. Now it is BDG. So we have not taken out the group from the name of the organization. The reason being that we want to stick to those values for which we have started this organization. And we started this organization in the form of the group. Now, what is the reason? Just see to this. We've created a group for students, researchers, scientists, and company professionals who are who are related to the field of bioinformatics, biotechnology, and pharma. So this group we created was on Google. What was the need of starting the group? Now, the need of starting the group was that the talent is definitely there. However, when the opportunity, you consider yourself, take yourself back in 2009, when the situation was that the opportunities were there and even the talent was there. However, by the time the opportunity reaches the talent, the opportunity is gone. So there was a time gap, or there was a gap between the information being transferred from the opportunity till the person, till the talent. So that is the reason, to fill this gap, we have created the group where we provide free consultation, tutorials, books, projects, and project reports, assignments, and latest updates for trainings, job offers, that is the most important in India and abroad, research projects, and latest science and technology news, and much more. So this was the reason why we have started this organization in the form of group, and till now it is there. Now you will be amazed to know that as of now we have more than 1 million subscriptions, and this is the number which is of September 2020. And these subscribers, they are not students, they are faculties, lecturers, scientists, and company professionals. So what we started in 2009, we are still doing. Subscriber platforms, and you could see that we have on social media, we are everywhere: LinkedIn, Facebook, YouTube, and Instagram, Twitter. So this is the subscriber thing is definitely the, you could see what's the percentage. Then you could see that on Messenger we have like BotIM, Google Duo, Telegram, and WhatsApp. And then we have other platforms like Pinterest, Flickr, and then you can see we have our own website subscribers. We have a join us page on our website where you could see that how exactly what we are doing, and you can join and subscribe to our newsletters. So we have a special and a dedicated team who works on this, what I should say, on social media platforms as well as WhatsApp, where people post their queries, and when they post their queries, the team is there to help them out. So this is what the subscriber platform. Then what exactly the company? So the company BDG Life Sciences, formerly known as Biodiscovery Group, it is its expertise in delivering quality services to biotechnology, bioinformatics, and pharmaceutical companies and clients. We started our operations in 2010, and the objective is creating a truly world-class organization which is fully, committed to offer best services to the industry. So I'll just go back to this slide. There you can see that we are, the wide array of our services include workshops, e-workshops, research projects, internships, training, webinars, seminar, and conferences. So this is what we have been doing since 2010. Then you could see that you have these bars. Now these bars will tell you that what kind of business we are doing. We are more into research projects. The reason is that this is the program which strengthens your profile. So we launch novel research projects on which people from different parts of the world have worked, and they are currently working, and you can do these research projects as your major project also, so that you can have much better opportunities. How? Because these are on novel topics, and most importantly, I should say that it will have an international publication by your name. So this means that if you do this, or if you do these projects, if you want to strengthen your profile, then you can do these projects. Then we have workshops. Now the workshops we have been doing, these are the workshops which we are doing on site. Like many countries, we have conducted our workshops, and in India as well as in other countries. So I just stopped my screen sharing. The reason is that why I have stopped screen sharing is that people are still coming, and I don't know why my team is allowing them. They are, someone is saying that I can't hear anything. Can you please type in and tell me if you can hear? Can you hear me? Yeah, Mariam, Bhagyashri, they all are saying, so how can Shima Ibrahim? Shima, please.
Yes, we are hearing you.
Yeah, so Shima, please check your internet connection, because I think that it's a problem over there in your internet connection, and that is the reason why you are unable to hear me. So let me again, start my screen sharing, so that, just hold on, please. Okay, so I will share my screen. I was on this slide. Okay, so over here, I was telling you about research projects, then workshops, which we have done in, say, like Dubai and Egypt and Mexico, Iran, Sudan, Nigeria, Kenya, Malaysia. A number of places we have done our these on-site workshops. In India, we have worked regularly at top institutes like All India Institute of Medical Sciences, New Delhi, then IIT Guwahati, IIT Vellore, and NITs. A lot of premier institutes have invited us, and we have been conducting regular workshops. However, since COVID-19 pandemic started, everything got stopped, and then now we have suspended all our on-site workshops till December 2020. However, we are planning to do the on-site workshops in 2021 if everything goes well, and we pray that everything should go well, and we hope that all people should remain safe. Then we have e-workshops. Now, e-workshops are the online workshops which we have been doing. A lot of online workshops we are doing. If you go to our website, as soon as you will click the workshops page, you will have two sections, because online workshops we are doing since 2012. And the reason why we have started online workshop, because a lot of international participants wanted to attend. And then we have started these things in the form, previously we used to do on a Skype, and then we shifted to Zoom, and then Google Meet. So the bar of e-workshops is a little higher than the on-site workshops, because a lot of people from all over the world have participated. Then, as I have already told you, that we have started the courses. Now these courses, we previously we used to do 4 or 5 courses in 3 months. The free courses, I'm talking about the free courses, just to give it back to the society. However, since this COVID-19 pandemic started, as I've already told you, we conducted more than 70 courses, in which more than 15,000 people, 14,000 people have participated from all over the world. Then we have others. Others is like contract research and project designing and all that kind of programs. Then what is the mission? Our mission is to provide the solutions for future by catalyzing the process of biomedical sciences into therapeutic technologies that benefit human health and society, and eventually fuel economic growth. Now this is what we have started doing, and this is the reason of starting the organization. So as an organization, we continue to work in the direction of growth as well as expansion, with an aim to serve more customers and make a difference to the society with innovative research work, employing latest technologies and excellent services. So this is the motive of the organization, that we want to innovate, we want to be creative, and that is the reason why we have been doing this. That is the mission of the organization. Services which we are offering. Now these are the topics on which we are offering our services. Now services, I hope you remember, we have contract research, we have workshops, we have research projects, we have courses. So these are the four kind of, and seminars. So these are the topics on which we are doing. So if you want, you can take the screenshots, or you can just go through our website, you will see. However, there is something very new in this, which we have started now, that is artificial intelligence and machine learning in biology, which we are using for drug discovery. Now drug discovery is definitely an important task in a biotechnology sector, as well as in the field of life sciences. So as we are shifting and the technology is advancing, hence we are moving towards it. And however, we have conducted certain courses on this particular technology, as well as now we will be launching soon some research projects. Then we have cancer, HIV, diabetes, and tuberculosis research. Most of the research projects which we have done till now. Now, these are the projects I would like to tell you that, the projects, the research projects I'm talking about, of which we have launched, and people like you from all over the world have worked. So these are the research projects. The number is more than 60, and all of them have got international publication. And people like you have worked, and their training sessions have been conducted online. So you can visit the research projects page of our website to get more information. Then we have the most important thing, and the most important thing is that why you want to get connected with BDG, why you should get training with BDG Life Sciences, why I should say that you should come to a place called BDG Life Sciences. That is the reason why this slide is here. Value proposition. Now what we do is, first we need to understand innovation. Now innovation is to translate ideas into service that creates value, and that is the reason. Now you can pick from this particular line, innovation is to translate ideas into service that creates value. So this includes some very important and heavy weighted words: innovation, idea, service, and value. Innovation is the core part of BDG Life Sciences, and why you will come to know why innovation is the core part of BDG Life Sciences, because we have been doing or pacing up the technology which is being used in US and Europe by launching programs which people have not even thought of. Like for example, artificial intelligence in drug discovery. So why you want to get connected with us? Why you want to get training from BDG Life Sciences? Practical application of technology. We learn by doing the things. Now I will move a little fast because I've already taken 26 minutes of yours. So I will not take much time. Practical application of technology. We learn by doing the things. So until unless you are not performing, you are not learning. So all our workshops, all our research projects includes practical application. Means that you have to perform. You have to perform on your own computer systems. Second, hands-on training sessions. Whatever training which we conduct, whether it is on-site, whether it is online, it has all hands-on training session. Even in silico molecular biology and genetic engineering or genomics and proteomics, we have hands-on training sessions. Novel research projects, I've already told you about that till now we have conducted more than 60 research projects, and right now there is one project on anti-cancer. You can view the research projects page. Then the most important thing, result-driven research. If your research is not result-driven, then unfortunately, you will not get the results. So result-driven research, and then the most important thing is everlasting relationship. I would like to tell you that people who have done research projects with us in 2010, they are still connected with us, and we are still helping them because they just keep on posting the query and we keep on helping them. However, one more thing, the benefit is that the person who has the research project, the person who has joined the research project in 2010, for example, that person was doing PhD, but right now that person is doing what? He has become a scientist. He is working for a good company, and still we are connected with him. So the person who will join now, we can recommend that person to him because that person is still in our in our circle. So, and that person who has become the scientist now, that person knows what kind of quality work we do. So they fully trust on the people whom we recommend. So that is the most important thing, and that is the circle. For example, if you join any of the programs with us, you are doing, say, MSc or M.Tech right now. After 10 years, you are still with us, and we pray that you do very well in your life. So you become what I should say, you become a scientist. What you will do is, that what is the quality of work. So after 10 years, if any student we want to recommend to you, you will get that experience. So that is the reason how we have recommending our participants to the scientists. Achievements, I will not go into this, but you can see that until unless this graph is going up, we do not have the problem. Now, the reason why this graph is going up, like for example, you have a great idea, you start with the great idea, definitely your graph will go up. But then if you will not innovate, or if you will not keep on innovating, then definitely it will, at one point of time, it will get saturated. So once you can see over here that this graph is not saturated. The reason is we kept on innovating. Mohammed, mute yourself. Yeah, don't unmute, please. Okay, because it's creating sound. Okay, so why this graph is going up? Because we have kept on doing something innovative and creative. That is the reason why this graph is going up. Then you could see this. This is, the summary of it, that 10 plus years of experience we have. We have the best of the trainers. These trainers, they give, we train them in such a way so that, they can give you the best possible training. 60 plus research projects we have already completed. That includes design, implementation, completion, and international publication. Then 400 plus hands-on workshops we have done in India. And just to summarize, 20 plus hands-on workshops in other countries, 50 plus hands-on workshops at premier institutes of India. And you can see over here that IIT, NIT, VIT, MMC, JITMER, etc. And then last, happy customers. Now, happy customers is we thrive on your comments because that makes us better. You suggest us, we work on it, and then we launch our next program. So feedback of our clients is something very important, and it is very, that is the reason why now we have started asking for the video feedback. So you can view our YouTube channel and you can see that we have posted the videos of our recently completed workshops. Now, as I have already told you, we were the first one to introduce online training in drug discovery and designing. We are also the first one to conduct training online and on-site in gene and genome editing using CRISPR-Cas9. And then we have trained hundreds of participants. I hope you know that we have already done this. So that's the thing. Free courses, as I have already discussed, that due to the COVID-19 pandemic, we launched free courses, and we've done for more than 14,000 participants in 70 free courses. These are the services which we are doing for scientists and PhD people: virtual screening, molecular docking, MD simulations, predictive toxicology and ADMET, NGS data analysis, and genome editing. Then these are something, two important things: personalized training. This type of training is introduced to help people having specific problems related to their research. Now, for example, we have a module. Now there are 10 topics in there, but then out of those 10 topics, you have 6 topics, but you want to learn 4 topics. So we will design a program having 4 topics, and then that is personalized training. Then research project consultancy is very important, because, like, for example, if you are a PhD student and you've stuck somewhere, then we give you the direction. I've already spoken to you about in the guides slide. So that is it, and you can just take the screenshot of this particular website, this particular slide, and you can see that how where we are available. And thank you very much. And now I will make or introduce Miss Sharvari Kulkarni, who is the technical associate at BDG Lifesciences, and she will now be presenting and taking the session for bioinformatics. So thank you very much, Sharvari. Now it's up to you, and you can unmute. Thank you very much.
Thank you, Steve, sir. Good evening, everyone. I am Sharvari Kulkarni. I am working as a technical associate in BDG Lifesciences, and I've done M.Tech in Biotech and Biochemical Engineering. First of all, my area of specialization is bioinformatics, in which I've done my research projects, and I have 10 plus years of experience of teaching bioinformatics, and I have certain international publications under my hat for the same. Now, first of all, I welcome you all to this session of bioinformatics, Introduction to Bioinformatics. And it's indeed a great pleasure to see overwhelming response and enthusiasm in all the participants to learn bioinformatics. And I assure you that these 2 hours will be really productive for you, not only to understand what is a basic of bioinformatics, to understand applications of bioinformatics, and you can also explore these basic things and tools which we will be learning in this 2 hour session to explore your research. So, uh, I would like to start with very basics because I think many of you are from biotech background or biology background, but some of you can be new to what the is bioinformatics. So I would like to start with very basics. Many of the times what happens is problem or there is a fear of bioinformatics. Why? Because of the difficult terminologies which are used in bioinformatics. uh, I would like to basically our aim of this current session is, and the module which I run for basics of bioinformatics is to make it as simple as possible to understand for the learners. So we start from the basics, and there are three stages of bioinformatics which we learn: basics, intermediate, and advanced. These 2 hour sessions will be entire focus on how bioinformatics growth was done, what is its importance in today's world, and what are the applications of bioinformatics. Then you can attend this session, and if you feel interest, we will be really glad. You can join our next intermediate and advanced workshops. So I would like to share my screen now. Let's get started with what is bioinformatics. Yes. So as you all can see, uh, life sciences is gradually being increased, or it is dominated by big data. Okay. So as it is expected that, if you can see here in this slide, it is expected that it started from 2000. Now we are here, and till 2025, it is expected to give at around 2 to 40 gigabytes of data. And that will be only the data of human genome. So you can imagine how huge amount of data can be generated with the help of life sciences. Okay. Now, in order to handle this data, it is extremely important to make sense of this complex data. Data is getting generated. Nowadays, it's getting generated at huge amount. It's because of pandemics like corona. Now, in order to make sense of this data, this complex data, team of interdisciplinary researchers is needed, right? So this interdisciplinary researchers means what? Researchers from biology, computational biology or computer science, and then statistics. Now, the pace by which this data is getting increased was not as fast as it is in today's world. So, there comes an importance of bioinformatics. So, it is the most emerging area, you can say, in today's world. We call it as a dry lab. What is wet lab and dry lab? Wet lab means your experiments. Now, we get data from wet lab, and then we use it with computer systems, with computer applications, tools, and softwares and databases in order to get results which are important to wet lab. So that is how this wet lab and dry lab are coming together. Okay. So now, if you see here, the next slide, I would like to move and give some basic definitions of what is bioinformatics. It is basically the computational branch of molecular biology, and it is multidisciplinary branch with vast applications. What do I mean by multidisciplinary? It is mean as there are many disciplines which are connected with bioinformatics. So it takes help from multidisciplines as well as it gives help to many disciplines. So that is how it is connected to different disciplines in order to give vast applications. And to go a little bit specification, I would like to define bioinformatics as conceptualizing biology in terms of molecules and then applying informatics techniques to organize the information associated with these molecules on a large scale. It works as 3D structure, sequence, 3D structure and function. So basically, data which we are going to get is only in the form of sequence. And we are going to use this sequence with the different databases, softwares, in order to predict the 3D structure. And from 3D structure, we can actually reach to the prediction of function. Now, as we move ahead with the session, you can actually think and imagine how vast or how brilliantly these bioinformatics techniques are used just from the sequence. How can you use it in order to get a lot of information and explore or give a direction to your research? So to present it pictorially, it is how it can be presented pictorially. You can see here, this is a living cell, right? So this is your living cell. And as you all know, if there is a perturbation or disturbance in the living cell, it shows dynamic response. Now, what are we going to do exactly in bioinformatics is we are giving this living cell, or we are converting this living cell in a virtual cell. What do I mean by virtual? Virtual means something which is not real. So we are creating replica of this living cell in the form of virtual cell. Now, this biological knowledge is in the form of computerized information. This information can be a sequence information. It can be a structural information. Once this information is given to the system, it is we will be calling it as a virtual cell. Now, this virtual cell is applied with some basic principles and we reach to very important and significant applications. Advanced part of bioinformatics is mathematical modeling. I will be highlighting that also. So this is how basically it is connected to different disciplines. So let's learn something about a little bit about the theory. Term bioinformatics was invented by Pauline Hogweck and Ben Hasper in 1970. Pauline Hagwick is a Dutch theoretical biologist and complex system researcher studying biological systems as dynamic information processing systems at many interconnected levels. Yes. Now you can see here this whole concept of bioinformatics. It started with, or it is initiated in 1988 as Human Genome Project. This is very important landmark of entire biology fraternity. Okay, so what was Human Genome Project exactly? Major goals in front of Human Genome Project was to determine 3 into 10 to 9 base pairs of nucleotides and to identify approximately 30,000 genes. So the computational task or challenges in front of this Human Genome Project were storage and indexing, building context, and scanning for genes. This is how graph of human genome progress goes. It started, as I told you, in 1988, and it was declared to be completed in 2003. You can see this finished part at around 100% in around 2003. Now let us see a little bit in detail about how bioinformatics emerged and when. Some major milestones of this particular field. So it started off with Watson and Crick, very famous DNA model in 1953. Then Sanger sequencing of insulin protein was done. Then at around 1969, you can say, or 1970, NW sequence alignment, that means Needleman-Wunsch algorithm sequence alignment. It is basically for global sequence alignment. Then at around 1969, ARPANET, which is early internet, was discovered. Then very important milestone that we will be discussing today is PDB, or Protein Data Bank, in 1973, you can say, was discovered. And in 1976 or 7, Sanger dideoxy DNA sequencing was discovered. Then NCBI, which is a major database. NCBI, this also we are going to learn in the session. NCBI is National Center of Biotechnology Information. This is also very important database. NCBI have major databases as well, which are sub databases of NCBI, as well as it has different tools like Map Viewer. It has tools like BLAST. Then SwissProt is also very important database that we use today also. Then FASTA and BLAST algorithm. Then World Wide Web. And in 2003, first human genome draft was declared to be completed. Okay. Now, let's move ahead with this slide I have put because I wanted to cover with most of the applications of bioinformatics. In this slide, I have covered all the applications of bioinformatics. Okay, so it is how it is related to different applications that we'll be discussing in brief, and then we will be going to the actual usage of tools and databases. Now, this bioinformatics is related, or it has an application of DNA sequence analysis. These many applications it has. We will be mainly focusing on BLAST and cluster. Then there are promoter analysis, gene prediction, regulatory elements. If time permits, at the end of session, I'll be giving one-liner description of all this. But we will be mainly focusing on main important applications, that is BLAST and cluster. Then very important application, molecular dynamics simulation. In that, you will be discussing about simulation, why it is important and what is the meaning of simulation. Then we'll be discussing very important aspect in today's world of bioinformatics. It is drug designing. So in drug designing, these steps we are going to discuss: target identification, target validation, lead identification, lead optimization, and ADMET prediction. Then protein sequence analysis, where we are going to discuss some primary structure analysis tools. So basically from where it starts, from where the information of sequence is used, and we start with our analysis. Then we are going to also learn how to construct phylogenetic analysis. What do we mean by phylogeny? Phylogeny is the evolutionary order with respect to time evolution as well as relatedness between the species. We'll see one example for each of this. I have tried to cover one example so that it is easy for you to grasp actual application of everything. Then you have molecular interactions, which will be discussing docking in that. So you have different types of docking. We will be discussing in about protein-ligand interaction or protein-ligand docking. Then we'll be discussing molecular modeling, in which I will be highlighting two tools for structure prediction and function prediction. Okay, so this is overall picture of what are we going to do in this 2-hour session. So let's actually start with what are databases. Okay, so let's start with what are databases. Databases are basically libraries of life sciences information which is collected from scientific experiments, published literature of high throughput experiments. They are, so database is nothing but where we are going to store all this information in ordered or organized form, so that we can use it with the help of different softwares and tools and interpret this data. So in bioinformatics, so many online and installed softwares are there, tools are there. So it is, of course, an important step to retrieve or use the data, but most of the times algorithms are set to the softwares and tools, and you just have to put your data as input query. Tool will do everything for you. There is no pay, and you will get a data. But to analyze and to interpret this data is very, very important. So database plays very important role where your information is properly stored. So they are broadly classified as sequence database and structure database. So these are some databases. These are some few databases which I have tried to include. These are all the databases, but we will be highlighting or we will be learning uh, 3 databases which I've selected because those are used commonly. Those are UniProt or SwissProt, PDB, and NCBI. Okay. Yes. So let's start with NCBI first. NCBI is basically National Center of Biotechnology Information. So this is the home page of NCBI. And here in this home page of NCBI, you can see you have options of all databases. Then you have many databases which run under NCBI. You can select particularly according to your purpose. Like in NCBI, there will be genome, protein, nucleotide, non-redundant databases, PDB. So it depends on you where you want to particularly perform the, or where you want to actually get the navigation. So first thing that you should know is how to use this NCBI. So here you can select option of genome, protein, and then you can simply put your query in the search. Okay. So this all actually we take it in our modules as a separate hands-on training and exercises, but we have only 2 hour sessions. So you try to understand with the help of screenshots what I'm teaching, and then you can, of course, if you want to go into more detail and explore more about it, you can join our module. Okay, so this is our basic attempt or basic purpose of this session is to highlight you with the main points, and then you can you can decide your area of interest and explore more about, because all of this, it is not possible to cover in 2 hours. All of them are a different topic and a different session of 2 hours at altogether. Okay, so now in the query, if you put your search, like here I have taken an example of insulin. So here I will select a gene or protein in the drop down menu. I will type human insulin in the search box, and this is how I get a page of human insulin. Okay. Now here, can you see all the information in a single go of just typing insulin in the search box? You will get all the information. You can later on, as a part of your interest, you can explore this homepage of NCBI. I'll tell you main important points which are needed to be covered. So if you can see here, there are 3 important highlights here. One of the important thing is FASTA. What is this FASTA? FASTA is a format of a sequence which is accepted by all the tools that we are going to learn. So what is the sequence? Any sequence of a protein is a sequence or string of course of amino acids, isn't it? So it is a sequence. Now, what do I mean by FASTA sequence? FASTA sequence is basically a sequence which is standard sequence accepted by all the tools. So for your idea, for as an example, I have used one Word file which gives you information about FASTA format. You can see here. So it starts with greater than symbol and the species name, or sorry, this is the accession number. So what is the accession number? Accession number is basically, I'll give you day-to-day example. Now, if we make an analogy of this, our classroom of any school will be a database, and every person in the classroom, if we decide to assign with them with roll numbers, we do have roll numbers in our classes, right? So like that, all the entries in the database are given one roll number. So accession number is nothing but a roll number. Okay, so key technique I will tell you of learning this bioinformatics is, there are many times why people have fear of bioinformatics is because we use complex terminologies in bioinformatics. So people who are hearing it for the first time thinks that it is something very great. So accession number, we get like, what is accession number? We tend to think like that, but it's simply a number or entry which is given to a protein. So it is simple and interesting if you learn it in a simple way. So this is an accession number, then it is followed by species name, then or protein name, and then species name. So here this first sequence, can you see it starts with greater than symbol, then it is a gamma fibrinogen protein, and it is a species name. Now this is a string or sequence of amino acids. Now whenever we are going to use it in any tool, we are going to copy and paste, we are going to copy and paste this sequence directly into the tool, and tool will do rest of the things for us. But copy and pasting of this FASTA sequence correctly is very, very important. Again, one more important thing in bioinformatics is you have to be really vigilant. That means you are not allowed to do typographical mistakes or just mistakes which are done by negligence. Like for example, here you are entering this FASTA sequence, and if you miss this greater than symbol, if you miss this space or you put additional space in between, tool will not accept it, and you will not even get to understand why tool is not giving me my result. So we tend to go to something, some reasonings behind it, and uh, the reason is simple, that you have made some mistakes. Okay, so you'll have to be very careful. This is about the FASTA sequence. Now, this was little bit about NCBI. So whenever we are running any tool that we are going to learn further in session, we are going to copy and paste the sequence in FASTA. That is one of the important thing that you should know. Now we are going to discuss about very important database, that is PDB, Protein Data Bank or Protein Database, right? So what is this protein database? It is the database which is a structural database. It is very, very important because it saves coordinates of proteins. NCBI gives you information about sequences. PDB gives you information about structures. So if your protein is a known protein, its structure or coordinates will be already stored in the PDB in the form of PDB files, right? So here how to use PDB is again very simple. You have to just enter here the search term. For example, you can take the same example, human insulin, or for I have taken here example of SARS-CoV spike glycoprotein. So what I'll do here is I will just put SARS-CoV protein name, and it will run the PDB database for me, and it will get me this entry which gives information about spike protein, as well as this is something which is very important, 5WRG. This is again same as an accession number in NCBI. 5WRG is a PDB ID code. It is always a 4-digit code, you can say. First, most of the times, first will be a numerical number followed by capital letters. Okay. Now, here, if I know PDB code, many a times when we use frequently these databases, we get to know, or we get to remember this PDB code. So you can enter the PDB code and it will take you directly to the search and retrieval of the data. If you don't know PDB code, you can type here your query and it will take you to the PDB information. Here again you have option. There are this is the page where you will be directed. Here you will be having options of display files and download files. So in the download files, you can see here again you have a facility to download FASTA, to download PDB format. Again, common mistake done here is you will have to download PDB format whenever we are dealing with any visualization tool, any docking tool. This PDB file we are going to come across very often when we are going to study bioinformatics. So PDB format this is a .pdb format. So you just have to click on PDB format, and at the leftmost bottom you can see your PDB file getting downloaded. So you just have to show it in the folder and save it at any convenient location in your system, right? So when you are saving it at any convenient system, convenient location in your system, you can simply use it for any particular application. Okay. Now the third database that I would like to discuss is UniProt. UniProt will also give you a FASTA file of the sequence. Same way you have to enter in this box your UniProt entry, like the name of the protein. Same I have taken SARS corona protein, which is spike protein. The UniProt also has its specific accession number. Okay. So if you know the accession number, you can put accession number. If you do not know accession number, you can put your query name here. Why UniProt is important? Because it is manually annotated and retrieved database. That means actually biological researchers work on this database for its manual annotation and curation. So the database is faster and it is highly curated or annotated. So this is how it gives you in very simple, or you can say in concise form, in a highlights. It highlights the function of the protein. Here again, you have a facility to get this FASTA format. So in the format, you go and click on FASTA canonical. You will get its FASTA file. Then you can copy and then you can use the same as we discussed before. What is FASTA? You can use the same FASTA sequence for different tools and databases. Okay, so these are the three databases which we have discussed by far: NCBI, PDB, and SwissProt. I believe you all are clear with these three databases. If time permits, I'll show you one example of each. If not, as I told you, you can just navigate through the database. And of course, as sir has already told you, we will be in continuous touch with you all. So you can put any query, you can put an email. We will guide you through usage of all the databases, softwares, and tools. Okay. Now, data analysis part. So we will be discussing, as I told you, primary structure analysis, secondary structure analysis, tertiary structure analysis, function prediction, and sequence analysis. So let's start first with the primary structure analysis, and it is the technique. There are many tools which runs for primary structure analysis. Expressy is the most popular and efficient server for this primary structure analysis. So first we'll be discussing prot scale. Prot scale is basically a system, or it is a software. It is very simple. It allows us to compute and represent the profile produced by amino acid scale on a selected protein. Now, what is this amino acid scale? Try to understand this first. Amino acid scale is any numerical value which is given to each type of amino acid. Okay, so that means depending upon the property of amino acid, literature survey has been done on around 57 such properties or 57 predefined scales, and those scales are used in order to decide which property is concentrated at which particular position in the protein. Okay, so the most frequently used scales are hydrophobicity, because hydrophobicity plays very important role in the functioning of the protein. So here, how to operate prot scale? You just simply have to Google prot scale. It will take you to this page of prot scale. Here you enter your accession number, or you can enter here the sequence. Now here you should understand that sequence, I mean only by the with the FASTA sequence. So here you can enter your FASTA sequence, or you can enter your accession number also. Now once you enter it, you can see here there are about 57 properties or scales which can be used. Molecular weight, bulkiness. By default, hydrophobicity by Kite and Doolittle is selected. Why? Because it is most oftenly studied characteristic. You can check, you can just click on any check button here, and then you can select that particular character. Now there might be a question in your mind why there are different names of after this hydrophobicity. It is because, or it is after the scientists who have discovered the scale. So we follow the standard scale of Kite and Doolittle. So it is selected. Once you click on that, you just have to click on submit. And this is how beautiful graph of hydrophobicity is obtained. Now, as I told you, We don't have to do anything, but you should understand what is the concept or how the tool is doing it for you. I'll tell you in brief, it is known as a sliding window technique. So at a time, there are amino acids which are selected with the position. Now, the amino acid scale values of each are plotted at on x-axis, and scores or scale values for each of the amino acid is plotted on y-axis. Now, how do you read this result? Is you locate the peaks wherever there are peaks in, as a protocol, as a standard. This proud scale output I have obtained it for human insulin. You can do it for any protein of your choice. Okay. So what you can do is you can put the accession number of that protein, or you can get its FASTA sequence from SwissProt, NCBI, any database, and you can put it there. Now, once you do this, you can locate the peaks. Now, if you locate here, this highest peak I can see here is at this position. Now, if I locate it on the x-axis, it is around 10th amino acid. So, you can interpret the result in a way like the most hydrophobic region in the protein is at around 10th position of amino acid. Below that will be the position of 40th amino acid, and least possible hydrophobicity is at around 55 position. So this will give you just the guideline about hydrophobicity. Then of course this result you can assume bioinformatics goes that way. First, you will try to get primary analysis. Then you can, on the basis of that result, you can explore that particular thing for your further research. So this is the simplest way of getting primary structure analysis in the form of any particular characters. So like I shown you in this previous slide, here down you have options like secondary structure, helices, beta turns. So if I select there as helices, I will get the Prot scale in the form of helices, helical property. So wherever I see the peak, there will be region of highest concentration of alpha helices. Same is the thing for beta sheets and gamma turns. So like that, you can explore about its structure also. Right. So the next and very interesting tool, which is very like favorite tool in our programs also, is SPDB, Swiss PDB viewer. What is the purpose of this Swiss PDB viewer? It is a structural visualization and comparison of two proteins. It looks very beautiful and it is very user friendly. So you have to but get it installed on your system. I'll tell you how to install it if you all are interested to use it. I will put the links in the chat box later on. So how to install this Swiss PDB viewer is basically you can Google also and then get the Swiss PDB installed in your PC. It will always come with this symbol as an executable file. Okay, this star symbol. So when you execute this Swiss PDB viewer application, it will be something like this. So here I'll tell you in brief how you can use this Swiss PDB viewer and what are the applications of it. You can simply go to this file and open any PDB file. You remember we have talked about PDB file. .pdb file you have to select here and open. You can see here this code 1HBG. It is for one hemoglobin protein. So what I have done, I've gone to PDB. I've downloaded PDB format or PDB file for 1HBG, and that is nothing but hemoglobin of marine blood warm. You can do it for any protein. You can load the protein. It will look something like this. Now, when I open PDB file of this protein, you have these many options. So we can't go into detail of each and every option. I'll tell you just important things. So here you go to Windows and this, it will look something like this. Wait, I will zoom it for you a little bit. Yeah, so it looks like this. When you go to Windows and control panel, in the control panel, this is the control panel, okay? So where you can play around with the molecule, whatever change you make in the control panel, that will be displayed in this interactive display. So this is the, basically, this is the superimposition which I have tried to show. Can you see here? These are basically these two proteins, one HBG and one GDI. These two proteins, now what is my objective of this exercise is, I've selected two proteins, which is hemoglobin only, but from different species. One HBG is the hemoglobin from marine bloodworm, and one GDI is the hemoglobin from plant root nodules. So they are structurally similar, being a hemoglobin, but there will be slight variation. And both the proteins, as the name itself says, what is hemoglobin? It consists of two moieties, hem and globin. So in the control panel, you can see all the amino acids which are there. Also, at the end, you have hem moiety present in it. Okay, so you have different ways of operating this. First, I have downloaded two proteins, then I have removed side chains in order to visualize it in a better way. Otherwise, two proteins looks very complex. Here it looks so simplified and so beautiful is because I've removed side chains of it, and I have given two different colors to different proteins so that you can see one white color is one HBG and one GDI is blue color protein. So you can see how beautifully they have superimposed on each other. Also, red color is one heme moiety in one HBG and white color is another heme moiety in one GDI. So they are also superimposed on one another. And most important thing, after superimposition, you can check out for how well they are fitted on each other. So that is how, this is how you can do it. You can go to fit. Then there is the option of magic fit. Magic fit is nothing but crude superimposition of two proteins. And then you go to Windows and alignment. Can you see here this alignment? This purposely I have taken this view so that you understand. This is one HBG and one GDI alignment over one another, but this is just a crude alignment. Okay. Now this you have when you are using this tool, then you use this mouse and hover your mouse over this one protein. I have just shown it for TRP15. Okay, so on TRP15 you can go to TRP15 and you can check out with which amino acid it is aligned. So it gives you RMS of 4.11 and standard alignment is 4.8. If your alignment RMSD is less than 4.8, then that alignment is understood to be significant alignment. Now, what do I mean by RMSD? For those who are new to this terminology, I'll just tell you in brief. Root mean square deviation is nothing but the deviation in between two structures in 3 dimensions. Means after superimposition, how one atom is oriented with respect to another atom in two proteins. So when do we say that alignment is significant or good? Whenever your amino acid in one protein is aligned to amino acid in another protein with RMSD which is less than 4.8 angstroms. Here you can see it is already 4.11. Now this tool has been modified in such a way that you see this alignment, and for the same amino acid you can go to fit, and there is one more option of generate structural alignment. What is the purpose of this generating structural alignment? what is this what is the purpose of this generating structural alignment? It is basically the generating structural alignment is basically to furthermore reduce the RMS difference. That means after generating structural alignment, again you check the alignment. If for the same amino acid, if the alignment falls below 4.11, then it is a significant alignment, you can say. Now, after doing this structural alignment, one more time, tool will try to superimpose in 3 dimensions these two structures in such a way that furthermore there is good superimposition in between these two proteins. So if I try to align it with the structural alignment again, it will give me RMSD which is less than 4.8 and it comes to 2.2. So what I can interpret from this is the two proteins, two hemoglobins proteins, they show great superimposition with one another. So this was about Swiss PDB viewer. Now I would like to tell you something in brief about Rasmol. Okay. So Rasmol is also very simple to download. You can just Google it or I will provide you with the link. You can click on this Rasmol latest Windows installer. It will take just 2 minutes. I would like to show you how to use Rasmol. Okay. So that it is little interesting. So when you install Rasmol on your screen, it will be installed as Raswin. So what you can do is you just click on Raswin. It opens with the command line at the same time. Okay. So you can open its command line also. And you can simply open any PDB file that I told you. This is how you can load your molecules. And very nicely you can play with this molecule. You can have different displays. Like this display is called as wireframe. This is backbone. This is sticks. This application of seeing all this in different displays is you can use it for your paper publications. Whichever display you like, you can visualize that way. And one more application of Rasmol is it is very fast to give you basic information. If your protein PDB file, you can load the PDB file and you can run simple commands. I'll show you fast 2-3 commands which can be run on Rasmol, like show information. Only the condition of Rasmol is it is very simple and user friendly to operate. But see, in 2 minutes, you just got the information about its name, its classification, its PDB record, then how many groups are there, how many helices, strands, and turns are there, like that. You can show sequence. So you'll get entire sequence of a protein like that. So these are some basic applications. Only the condition of Rasmol is you have to be specific in commands. As I told you, you cannot put any additional space or you cannot skip the space. Okay, so I can zoom the protein also, zoom like 200. If I give the command, it will be zoomed. So like that you can. This is a primary visualization tool. Okay, so this is how you can play with this, or you can use with this molecule. Then let us go to the next part, secondary structure prediction. Yes. So I believe these two visualization tools are clear to you. Now let us go to secondary structure prediction. This was about visualization and primary structure prediction. Now let us discuss secondary structure prediction. There is very famous algorithm which is known as Chow-Fasman algorithm for secondary structure prediction. Chow-Fasman algorithm, basically it is a sliding window technique. What do I mean by sliding window technique? Sliding window technique is at the time you consider a window of particular size. Now again, the same thing I would like to tell you. Secondary structure prediction is also done by the tool. Chow-Fasman server is, I'll show you. This server is so simple that you just have to put your query over here and then it will predict and give you this result. But I would like to tell you how the algorithm actually runs. What do I mean by sliding window technique? So in the sliding window technique, you select a particular window size and uh, for, for example, P alpha. How do you locate alpha helices is for 4 out of 6 contagious residues, you scan the window and check out if P alpha, that is probability of presence, every amino acid has its probability of being present in that particular structure. So see here, you can go through this table. So each amino acid has its particular probability to be present in P alpha or alpha helices depending upon its properties. Its physicochemical properties decides in which type of structure it will show tendency to be present. So P alpha, if it is greater than 1 for 4 contagious residues, 4 continuous residues out of 6, then that particular area will be called as P alpha helix. Now you extend this alpha helical regions in both the directions and locate wherever region is P alpha showing greater than 1. Then next is P beta. 3 out of 5 continuous residues, if they show probability of beta greater than 1, then that particular region will be called as beta region. Okay, and it will be also extended as a sliding window. Sliding window means what? At a time, if I'm considering 6 continuous residues, I will mark that as a P alpha region or P beta region. P alpha region. Then I move ahead or shift by 1 amino acid, leaving back first amino acid. Now, next window, I will again try to locate out of 6 continuous residues, 4 residues if they show dominance of alpha helix. Again, I shift by one amino acid. Then I'll check out if it is showing probability of presence in the alpha helix, which is greater than 1. That way, you locate P alpha and P beta. So all the alpha helical regions and beta regions are located like that. And rest of the regions are located as turns. But now the formula has been developed for turns also, which is f of j plus 1, f of j plus 2, where j is the residue which is under consideration. So when we are considering turn, that time we are considering 4 continuous residues. Okay. So this is the Chow-Fastman server, into which you can see here, you can put your query over here. That means your FASTA sequence. You can go to predict and it will give you these nice results. So simple to read. So just the position of amino acids, helices, sheets and turns. Okay. So here you can see this is the query. So wherever we are dealing with any sequence in bioinformatics, so we'll call it as a query and database as a subject. So this you can see your query sequences. Then E are the turns, H are helices, and T are turns. There is one more region known as coils, C are coils. So this gives you exact position of helices, turns, and coils, as well as it gives you, you can see here at the end, total amount of residues are also given. So you also get to know the position. So as well as you also get to know the total number of residues. That means helices are 48, turns are 30, and beta sheets are 30, and turns are 8. Okay. So like that, you can read the result and understand about the secondary structure prediction. You can read it in the form of bars also easily. So this red bars are nothing but your helices. Then your blue bars are nothing but, green bars are nothing but your sheets, and blue bars are your turns, with the yellow bars, which are a little bit faint, are coils. Right? So this is about secondary structure prediction. Tool which is used is Chow-Fastman server. It runs on sliding window technique, and this is how you get the result. So as you can see, it is going in one order. First, we have tried to analyze its primary structure on the basis of different characteristics. Then we are moving with the visualization, and we are trying to compare whether any other protein is related to it. Then any other protein which is related to it. Okay. Then your secondary structure prediction. Then the next very important application of this particular protocol is sequence alignment. You must have come across this terminology very often, that what is a sequence alignment? What is a BLAST? BLAST is very famous tool in bioinformatics. So let's learn about sequence alignment. What do we mean by alignment? In a day-to-day definition, if you want to define alignment, it is a proper way of arrangement. It is a way of arranging sequences of protein, RNA, and DNA in order to infer structural, functional, and evolutionary relationship. So, by means of sequence alignment, what are we going to understand? We will be understanding structural by means of sequence alignment. If two sequences are highly similar to each other, Then their structural similarity can be interpreted, their functional similarity can be understood, and their evolutionary relationship or relatedness can be understood. Now, there are two ways of pair alignment, pairwise sequence alignment and multiple sequence alignment. Okay, pairwise sequence, there are basically two ways of alignment on the basis of technique. One is global and another is local. Global is entire length and local is only the short stretches of sequences. And based on the number of sequences which are considered at a time, there are two methods, pairwise sequence alignment and multiple sequence alignment. So let us see each of this one by one and each of the software which can be used for this two purposes. So I've tried to make it very simple for you all to understand. In practical applications, we don't deal with such short sequences. We deal with large number of amino acids or nucleotides. But here, for you to understand the concept, I have just taken four number of nucleotide sequences. So you can see here. So the first is ATCG and next is TCA. ATCG is aligned to TCA and the alignment is called as pairwise sequence alignment. The first position there is a gap, you can see. Now, if you want to study this alignment, just think how can you study? These two are matches. Again, these two are matches. This is a mismatch. So how many matches are there? Two matches are there and one mismatch is there with one gap. Here, if you say, if a person X is doing the alignment by this way, person Y can do the same sequence, use the same sequences and do alignment in this way. So here you can see in the second type of alignment, ATCG is aligned to TCA with the gap at the end. Now, if we study this alignment, how many matches and mismatches are there? This is a mismatch, this is a mismatch, this is a mismatch again, and this is a gap. So there are no matches. Here at least we are getting two matches. Here we are not getting any match. So logically, which is the best alignment? First alignment. So this is the purpose. Pairwise sequence alignment by BLAST is done in such a way that you have maximum number of matches. So the tool will apply algorithm in such a way that your number of matches are maximum. Now, sometimes it will produce gaps also, but that gaps are produced with the gap penalty. So, in order to get maximum number of matches, algorithm will learn and it will run and it will give us maximum matches. These matches are interpreted with maximum homology. What do we mean by homology? What is the difference between homology and similarity? Similarity means sequences are similar to each other. Homologous means they belong to common ancestor. Now, if these two sequences, they show great similarity, they belong to common ancestor. Now, going ahead on the same concept, I would like to tell you something about scoring scheme. Now, see here. We'll again consider the same example, ATCG and TCA. Now, scoring scheme can be defined by user. Here, the first example, can you see here? I have given this -2, 2, 2 and -4. So my cumulative score comes to be 1. For the same alignment, these are distantly related. For the same type of alignment, two distantly related sequences are considered. So we get a least score. Now in these two sequences, scoring scheme is score is 1 with this scoring scheme. So can you see here what is the scoring scheme? -2 I have assigned for a gap, then +2 for a match, again +2 for a match, and -1 for a mismatch. Now I would like to give you an analogy for this scoring scheme in simple words. It's like attempting a competitive exam. In competitive exam, if you have negative marking system, then what is the type of marking scheme you have? For exact same answer, you have +2 as a score. For related answer, similar answer, which is not a correct answer, you have -1. And for not attempting the question, you get -2. So gap is like keeping your question unattempted. Understood? So it's like that. So each algorithm has its own scoring scheme. Now this is the actual homepage of the BLAST. This is how it looks. BLAST is a search tool of local alignment. Here you have different flavors of BLAST. Like nucleotide, you can perform BLAST for protein also. Okay. So today I'm going to show you one protein BLAST as an example. So here I will go on protein BLAST. I'll select any one protein. Here I have used the same protein like which I use for prod scale. It is a gamma fibrinogen. You can see here gamma fibrinogen with accession number O12959. This is its Swiss prot entry, UniProt entry. So I have used that FASTA sequence and put it in the BLAST window. I've just run the BLAST and it will take me to this result page. This is the result page of BLAST. It gives me the this information, which is easily readable, but interpreting BLAST result is also a skill task. You can see here topmost entries are with the fibrinogens, of course, because it belongs to same family, but with different species. So the first most important hit or with the maximum score is with the 132 as maximum score, query coverage as 100%, and e-value as 2 e raised to -37 with the percentage identity 76.62 and accession number as P12799.1. So this is how you understand how many number of matches. So what is done here exactly is you have put your query sequence and BLAST has tried to connect to the database of NCBI and it has run or it has given you all the results which show this much score of similarity or identity with this much e-value, and it will tell you how many sequences are similar to your protein. So isn't it a very valuable information? You get to know significant alignments. So that means how can I interpret? Gamma fibrinogen of this particular species is related or is identical to this Bos taurus, Homo, that is one family of cattle, then Homo sapiens, Rattus, Mus musculus, like that. Okay, so generally we select first 5 alignments or first 5 hits to be most significant one. So you can imagine from a single sequence, you have reached to the point where you know which existing proteins are similar to your protein. Okay. Now see here, what is e-value? This is something must be new for you. Score and all you can understand. Maximum number of identities, nothing but your score. So what type of alignments are considered to be significant are those with the maximum score and those with the least e-value. Lowest is the e-value, most significant is the alignment. Okay, what is e-value? It is expectation value. That is your hit or your alignment has come if just by chance, then it is e-value. So those values or those hits which are having more E value, those are not considered to be significant. Here you can see 2e raised to minus 37. How less is that E value? So you can say that this first alignment is significant. Understood? This is about the tabular form of information. Then can you see here? These options are there. So graphic summary, if you click on this graphic summary, it will take you to this page where you have beautiful bars which represent your information. So you can just read this graph, you can understand. Here, pink bars which are represented, they show highest scoring alignments. Green lower than that, blue lower than that, black we don't even consider. Usually if the percentage identity is very high with the protein which you have selected, you will get red bars which are greater than 200. But here unfortunately we don't have scores which are greater than 200, so we don't have any red bar present. If you select, you can do it for any other protein of your choice. So if you do it for protein like collagen and all any other protein, you can type in the blast. You can see the red bars if you have maximum score which is greater than 200. So this is a graphical display of your protein's similarity, your query's similarity with the database. This was about the pairwise sequence alignment. Pairwise meaning two sequences are considered at a time. Then moving ahead, I would like to discuss about multiple sequence alignment and phylogram. What is a multiple sequence alignment? Multiple sequence alignment is at a time you consider more than two sequences. Okay. So like the same as the BLAST algorithm is the same, but at a time you are considering more than two sequences. And with the help of this multiple sequence alignment, here also we are going to use faster sequence, but more than two sequences. Now, if you are giving this tools which are used for multiple sequence alignment are cluster W and cluster omega. So in cluster omega, you have at a time more than two sequences. You just put the sequences in cluster omega and it will give you the multiple sequence alignment. Now with the help of multiple sequence alignment, you build phylogram. Phylogram is again a very important interpretation result that you can get. With the help of phylogram, just by reading the tree, you will understand how the proteins are related to each other, how the species are related to each other. Going ahead in the phylogram, there are sometimes branch lengths which are scaled to time, evolution, and relatedness. Okay. Now, there are some important terminologies which I would like to make you familiar with when you are learning phylogram. Root, node, clades. With one example, we are going to see. Root is, wait, I'll show you the phylogram and then we will discuss. It will be more easy for you to understand. So this is your tool, cluster omega. In that, can you see here in the query box, I have put all the cluster sequences which I had shown you as a word file. All those sequences simply I have copied together and pasted. Then you simply run multiple sequence alignment. This is how your result looks like. You can show it by colors. Here you have, where I have this hide colors, there you have show colors option. You just have to click on show colors option and then you will get multiple sequence alignment of the sequences. So I have taken only 3 species for you to simply understand, but at a time you can do up to 20 to 25 sequences also. So this is first sequence, you can see it is gamma fibrinogen of chicken, second is human, and last is chimpanzee. Color indication shows you that this particular amino acid is conserved throughout. Okay. So star symbol or asterisk symbol over here gives you information about its throughout conservation in all the species. Can you see here? At some places there is two dot. At some places there is one dot. Two dot in terms of multiple sequence alignment means semi-conservation. What do I mean by semi-conservation? Semi-conservation is when amino acids are not identical but they are similar. Means depending upon the properties, they are they belong to similar groups but not identical groups. Okay, so that times your two dots come. That is called as semi-conservation, and one dot means there is slight conservation. They belong to different groups. So this is how you can read the multiple sequence alignment. Now here you can see option of phylogenetic tree. You can just click on this phylogenetic tree and you reach to this result page. Now let me explain this in a little bit detail. You can see here this phylogram. This is also called as a cladogram. I'll tell you what is the reason behind it. Can you see here? Now how to read this phylogram? This particular place from where branches are splitted is called as a root. This particular point where the branch which comes from root is splitted furthermore is called as a node. So if you want to read this tree, how will I read? Human and chimpanzee protein being present on the same node, getting splitted from the same node, having equal branch lengths, they belong to same clade. They belong to same group. So they are highly similar to each other. So their similarity score will be also higher. This is why they are called as one clade, and that is why this type of tree is called as cladogram. Now this node is also related to the branch which is related to chicken. So that means chicken protein is also related to human and chimpanzee gamma fibrinogen protein, but at a larger distance. Why? Because it is coming from the same root, but it is present on the different clade, and it does not show equal branch length with these two. Understanding? So that is how you read this phylogram. I'll repeat once again if you don't understand. This is one clade because it shows equal branch length from this node. This tree has only one node because we have selected only three. Now there can be 2-3 nodes. So whenever you are reading tree, that time you read it from root and nodes. This is one node. There can be 2 nodes, 3 nodes, depending upon how many clades you have. So since it belongs to same root, these three belong to common ancestor. That means they are homologous. But the similarity percentage in human and chimpanzee is more because they are present on the same node with the equal branch length, and similarity score with the chicken is less because it is present at higher branch length and on a different branch. Okay, that is how you read phylogram. Now phylogram is also a different topic of research altogether. So when we are doing this in our workshops, we do it hands-on. Participants, they do construct their own phylograms with the exercise of around 10 to 15 species or their own proteins of their choice, and we tell them, or I give them exercise to interpret this results. Okay. There are two types of phylograms, rooted and unrooted. This kind of phylogram is called as rooted because it has root. Sometimes they are radially sprayed. There they are called as unrooted. Okay. Now this was about the phylogram. Now let's move ahead to the next tool. Our purpose is to cover up maximum tools, but let's see how much time permits. At the end, we'll be having question answer session also. So I just hope that I'm not running too fast and you all are able to grasp what I'm saying. Yes. So next is interpro scan. Interpro scan is basically, so as you all can now just go back and revise whatever we have studied: primary structure analysis, secondary structure analysis, superimposition, secondary structure prediction, and comparison, blast and cluster. So till now we have reached a point where we can, at least we are in a position to understand which proteins are related to our protein. Now you can go ahead with different tools to predict the function of the protein. So InterProScan is a very beautiful tool and very applicable. It has vast applications. Single page gives you or redirects you to so many databases that on a single go, you understand which functions are associated with your protein and which all families are associated with your proteins. That is very important information that you get in InterProScan. It is a domain function and family function prediction. This is how Interpro results are obtained. Interpro is also very simple to operate. You just have to type Interpro scan in Google and you can paste your sequence like the way we have pasted, same FASTA sequence. Okay. Once you paste the FASTA sequence, you have to click on Interpro. It will give you this kind of result. Can you see here these bars? So function prediction, so simple and so clear. It will, since the same protein I have been using throughout, gamma fibrinogen, its biological function is given as platelet activation, protein polymerization, blood coagulation. If you click on this particular entry, it will take you to the research paper associated with it. So it will give you all the information about that function also. Then its molecular function, its cellular component. The most important application of this Interpro scan is, as I told you, it will tell you classification of protein in terms of families and domains. What are domains? Domains are blocks which are assigned with a particular function. So any particular block or repetition of the same amino acids in the form of domain is repeated throughout the species. So if it is repeated throughout the species, it tends to give you the same function. Okay, so if you can actually do the exercise and see, when you click on this particular entry, it will take you to the database of that particular database. So this is iProInt. So it will take you to the Interpro database and it will give you information about its function. Next, you can click here PTHR. It will take you to the Panther database. Panther database is a specialized database for gene annotation. Then like this, same way you have different database and redirections to the database. So if you click on this SMO, it will take you to the smart database, which gives you information about the architecture of the protein. Architecture means what? Its helices, again, its tertiary structure, its secondary structure, all that. So this is how this particular information can be obtained in one go with redirections to so many databases which are specially designed for that function. Then there is one more important thing which is included in Interpro is disorder function prediction. This one. So it also tells you the area which it undergoes, or if it is disordered. Disordered area means disordered structure, and which will have impact on the functioning of the protein. Okay, so this is one tool that we learned about function prediction. Now there is one more tool, FIRE tool, which is also very important tool, and again it has many applications. By using sequence in one go, you can get lot of information about its domain, about its template. Then recently it has been introduced as FIRE to Investigator. FIRE to Investigator gives you one very important application of sequence profile and mutation profile. Okay, so this is how your FIRE2 result comes. Will not go into detail of this domain and template analysis. You can do it for yourself. It's very simple. It will just give you the domains and its information template. What I would like to focus on is something which is sequence profile and mutation profile. How to read this by usage of Pirate to Investigator. You can run it on your own. Pirate to Investigator, after it's run, it will give you this result. Now, this is again a very simple to operate, but then you will have to interpret and study results. These two tools, Interpro Scan and Fire2, are very informative, but you should be good to use the database. That is also a smart and skill required technique, I would say. So here there are a number of options and you can explore as much as you want. Okay, Ramchandran plot is also there. Alignment confidence, that means how much confidence or accuracy is there with the tool. Now, I would like to explain about this sequence and mutation profile because it is very important from research point of view also. So, I get the secondary structure. You can see how beautifully it is depicted. This green are alpha helices and blue are beta turns. Okay. Now, if I move, this is the query sequence. If I move my residue, move my mouse or scroll my mouse over these residues, this I have highlighted for F. Okay. Now, if I highlight or if I keep this mouse on F, can you see here these two types of graphs are there? One is sequence profile and another is mutation profile. Sequence profile is basically a graph of probability of amino acid at that particular position. So the tallest and generally color depictions are given for all the results in this Pirate 2. One nice thing about it is it always gives you color depiction. So blue is always good and red is always bad. In this type of depiction, this is a JSmall view. Okay, like a Rasmol, JSmall is basic visualization tool. But here, red is the highest confidence level. You can see here for F amino acid, it's phenylalanine. You can see here in the sequence profile, tallest bar is present at F. So that means probability of occurrence of F at this position is highest. Probability of occurrence of F at this position is highest. You can see here. That means, and blue and green are the lowest. So F is the highest probability amino acid at that position. At the same time, if I read this mutation profile, at the same time, if you see this mutation profile F, red bar with the tallest height is seen. Can you see here this red and yellow bars? What do I mean by mutation profile? Listen very carefully. Mutation profile is that position of amino acid which, if replaced by a particular amino acid, shows major impact on the functioning of the protein. That means that particular mutation is significant. Now, if I see here, F is present over here. If I check out in this mutation profile graph, tallest bar is visible at G. That means if F is mutated by G, it is the significant mutation. That means that type of mutation will definitely have some effect on the functioning of the protein. Lower than that will be this bar. If I'm not wrong, it is for E. So if F is replaced at this particular position by E, it will have mutation which is significant at that particular position. Are you getting it? So this is how you can get basic idea. There are again modified tools, and mutation analysis is not a very easy task, but this gives you basic information about where and how mutation is affecting your protein, right? Now we come to the very important module of this session or this bioinformatics, which is nowadays very like you can say important and applicable because of the time which is spent in the wet lab and expense which is spent in wet lab, and that is nothing but your drug discovery with the help of in silico. In silico means what? With the help of computer. So this is drug pipeline. These are the steps. Disease related genomics is studied first. Then let us take one example for you to understand the protocol. Very common disease, cancer. Now, if you're working on cancer, then first what will be your like protocol for designing a experiment? Cancer is your target disease. So you'll first study the disease related genomics. Then what will be your target? Protein which are anti-cancer, protein which are receptors. So we can talk like topoisomerase. Topoisomerase, what is its role? It has its function in DNA replication. So if topoisomerase is inhibited, it will automatically inhibit the cancerous growth, right? So if uh, so what will be the role? What will be the task? To inhibit the topoisomerase. So there comes your target identification. So here your target will be topoisomerase protein. Once you identify the target, you validate the target. Validation is nothing but drugs targets, drug ability is predicted. Sometimes you need to validate the target in terms of their structures. You have to remove some unwanted chains. You have to add hydrogens like that. That target validation is done with the help of tools like Swiss PDB, which we have done. Okay. Then target identification is with the help of PDB. You can identify the target. Lead discovery. What do I mean by lead? Lead is nothing but your ligand, drug which you are trying to impose on your target. For example, uh, I'll tell you the example which I have done. It's actually we had worked on topoisomerase inhibitors. Like naptoquinone is one family of proteins. So like that, you select one chemical family and select some drugs or ligands out of it. That comes as your lead discovery part. Then you optimize the leads with the help of lead optimization tools like ChemSketch, Marvin Sketch. Then you perform docking studies or you impose receptor on ligands, check the docking score, and then uh, before docking, sometimes you perform simulation also. That's the best way to follow. You simulate the target and then you go for docking studies of it. And based on the docking score, you can come to very important results of their interactions with amino acids. We are going to come to that part also. We will be discussing that also. And then it goes to preclinical tests and clinical tests. So can you see here? You can just think how much part we have, how much time and money we have spent by using computational tools. Not only these tools are fast, but they are also accurate. Yes, so this I have discussed in the previous slide itself. Now I would like to discuss something about simulation. Simulation is basically something you make mimic of. Okay. So why simulation is important is because we are trying to mimic the system or our target the way it is present inside the body. Why we are doing so? Is because we are dealing with drugs, we are dealing with receptor proteins, and those are present in certain type of environment, right? We are trying to provide that kind of environment to proteins and ligand, so that we come to a point or data which is very closer to our natural data. What is the technique for that? It is called as MD simulations, molecular dynamics simulation. Basically, so it is nothing but analyzing the physical movements of atoms and molecules. It is the computational method that calculates the time-dependent behavior of molecular system. What do I mean by time-dependent behavior? That means for a particular molecular system, we are going to select one time step, like 2 femtoseconds, 5 picoseconds, depending upon the user-defined values. You select a particular time and check out the behavior of system for that particular time. And it gives you information about the fluctuations and conformational changes of proteins and nucleic acids, which are very, very important in order to understand whether we are reaching to the stable system or not. These type of methods are routinely used to investigate structural dynamics and thermodynamics of biological complexes. Okay. So, in short, what are we doing here? Can you see here this diagram? This system is made up of n particles. Okay. So, to this n particles, we are going to give certain kind of potentials, and we are going to give fluctuations to this system. There are 3 types of force fields which can be given: char, amber, and explore. Okay, so most of the times charms is used, amber is also used. There are different softwares which runs this simulation. NAMD, VMD is there, Chromax is there. Nowadays, many others are also introduced. So basically, simulation is also like an expert task. You have to correctly make the topology files for simulation to run. I'll tell you first the concept of this simulation. So are you getting it? So there are n particles you can see here. To this n particles, we are applying fluctuations, and at the end of all the time steps which you have selected, at the end of which we are going to see the result of simulation. And if our system shows RMSD which is less, we can infer or add the energy values which are less. So we all know least is the energy associated with the molecule, more stable is the structure, right? So we get to know if our structure is stabilized after simulation or not. And that stabilized or simulated structure is always a recommended structure, or it is always advisable to use a simulated structure, because it is a simulated structure which will be similar to environment present in your body. Okay. Now there are simulation also. We have a different 7 days workshop for simulation, in which I teach hands-on to how to prepare all the files required for simulation, how the topology files, PDB, PSF files are generated, how to configure it and make configuration file, and how to run the simulation. Simulation is run on the command line prompt, so that also is taught how to run the command and how to make your simulation run and how to analyze the results. That is also a very important task. So all the graphs are prepared, and you will be able to understand at each time step how the fluctuations are done on each particle and how your system has undergone the fluctuations and achieved the stability at the end. Okay. Next part comes as a docking. What do I mean by docking? In simple words, docking is to dock means to fit something. So here you can see here in this diagram, this is your protein. I have just zoomed or to make it more visible and clear. This is ligand and this is our protein. So this is the active site or pocket of a protein. These are the domains N, SH2 and CLO. Okay. Now, this ligand is fitting here in this pocket, and this is called as docking. Docking is the technique of fitting of ligand geometrically and energetically favorable. What do I mean by energetically favorable? Conformation of a ligand which gives least possible docking score. So docking score has to be least and interactions have to be perfect. Perfect meaning proper interactions with amino acids. That is called as a fit. It's like a lock and key model or your enzyme substrate reaction, right? One lock fits into one key. Same way, it will have one particular binding pocket where your ligand will fit one fit, and it will give you a proper conformation which is energetically favorable. So there are different tools for performing this docking. We use very standard tool which is known as Autodoc Vina, and Autodoc Vina gives you results in the form of 9 conformations. I'll show you. This is the picture of how Autodoc Vina looks like. Yes. So here ADT is basically you have to install it again. You will get it online. It is free. ADT you have to install with MGL tools. Autodoc Vina. Vina is executable file, but Autodoc Vina does not have its own GUI. You have to make changes here, prepare the files, and then you have to run it in the MS-DOS command prompt calling Vina. We also have a separate module of docking which I conduct for about 5 days workshop. So there I teach hands-on how to prepare all these files and how to do docking for single ligand, as well as we have included it for multi-ligand. So you can attend that upcoming module. You will be getting information on BDG website for that. So right now we will discuss it in brief, not hands-on actual, but try to understand and grasp whatever I'm saying. I'm telling you concept and usage of the tool. So now, as I told you, you have one receptor. Receptor can be your protein, anything. If I tell you one example of SARS-CoV-2, same. You can take this spike protein and load it here by opening in ADT as a file, open PDB file. You can get a ligand. You can get the ligand and prepare the grid box. So go to file, then you have grid box where you can prepare the grid box with XYZ coordinates. You will not get accurate grid box, but you will get at least the scales, and you can save the grid box and you can prepare the configuration file. Once you prepare the configuration file and run the command of Autodoc Vina, you will get a result in this form. A log file will be created in the folder automatically after the Autodoc Vina run. Okay, and that will give you affinities. Can you see here 9 confirmations with the affinity values? So lowest is the topmost, which is minus 6.1, with the RMS D value 0. So these first 3 confirmations you can select. Minus 5.6 is the decent. All the values which are above minus 5.5 can be selected. Those 3 confirmations can be treated as the best mode or best ligands of docking on your receptor. Then you can visualize it in any visualizer tool like Pymol, Chimera. Okay, this is a view of Pymol which I have taken. You can see how beautifully ligand is sitting inside the pocket of the protein. So you can see here, if you see carefully, 1 to 9 modes are given. This ligand can bind 9 at different 9 positions. Okay, like that. You have 9 possible confirmations, but you select only the first. You can cross check with the log file, and you can select only the first 3 models that are best possible fit of docking. You can visualize it if you have Discovery Studio, Chimera, like that. Okay. So like that, you get to know docking results, and that is how you can confer to very important information of which ligand fits best to the receptor. So this is about the docking. Then I come to the last part of session, and that is QASR. This is something which is very advanced part of bioinformatics, quantitative structure activity relationships. So here you are just having information about the physicochemical properties and topological properties of your ligand. You don't have information about the receptor. That time, how to go about the study or research is QSR. So you have some term which is known as descriptor. There are 2D and 3D descriptors. Then you build the model with, this is highly mathematical technique. I'll tell you in brief how to go about QSR. This is how first you have your molecular structure, then you have molecular descriptors, then multivariate analysis, correlation to activity, and then model validation. So basically you have 2D, 3D structures of molecules, and you have these type of descriptors. There are different tools like VLife, MDS, QSR build for QSR. You can see here there are different descriptors like constitutional, electronic, geometrical, hydrophobic, lipophilicity, steric. These descriptors are nothing but properties of ligands. So those properties are selected, and they are experimental activity data is available. So mathematical model with the help of multiple linear regression, PCA, NN, support vector machines, KNN, mathematical model is built on the basis of information related to descriptor properties and IC50 values. And that particular model is correlated to activity. Once the activity and structure relationship is there, that model can be used to design new molecules, as well as if your ligand has known descriptors, then you can predict their activity. So you can imagine how important role it plays. If you know what are the certain phytochemical groups, you can select the chemicals from that family, and you can put its descriptors values like hydrophobic descriptors, lipophilicity descriptors. You can select as many descriptors as you want, and their IC50 values, like 50% inhibition values. Make one model. Then next time when you are coming across with unknown ligand, you can just put its values in the equation and you can predict its activity. If it shows decent activity, you can actually design it in a wet lab. So vice versa can be also done. You are taking data from wet lab, you are also able to provide the data for wet lab. Okay. So there are some basic books which I have used for, which are put here for beginners. There are n number of books on bioinformatics. Indian author books, foreign author books are very good. And in David Mount, you will get information about sequence genome analysis. You have Gibbas, you have Bergeron, which is an excellent book on for those who want to learn bioinformatics. Also, I use Baxwenis, which is very nice book. You will get it on Amazon also. Then, if you are in touch with us, I will be happy. It's all my pleasure to help you with the links and books names. You can mail me at any time. You can get our contact through BDG website. That's it for 2 hours session. I believe that I have provided you and benefited, and your 2 hours have been productive. And if you have any questions, we can discuss.
Thank you, Sharvari, for a very nice and informative session. Now, I would like to see, like I am seeing one question is from Mr. Asif, and he wants to know what is the importance of energy minimization in drug design.
Yes, Asif, I would like to answer your question as Energy minimization, why it is done is because, as I told you when I'm explaining, system, when we are performing any task in silico on the system, that time the system might not be stable energetically. So if we minimize the system energy of the system, what will happen? It will give us a stable structure. So that is why when simulation is performed, first step is minimization and next step is equilibration. So that is the importance of minimization. Always remember, whenever system is attaining minimum energy, that time it is more stable. So stability is the significance of energy minimization. Okay, Asif?
Thank you, Ms. Sharvari, for the answering this question. Now, there is, as you know, that this session is also live on YouTube. So I have got a question over there, and Anuj is asking how to devise a strategy in case of identification of novel protein. So, as far as I understand, that if the protein structure is not available, then you go for homology modeling, because homology modeling itself
Yes.
is the technique which we use to identify the structure of the biological target, or I should say, model the structure of the biological target, that by which we model the protein on the basis of the template. Now, how it is done, as we all know that we go for the protein blast, and then we find out the closest related proteins, and then we make it a template, and then the target, and then once we model the structure of the protein, then we go for the model validation. Now, model validation we do for Ramachandran plot, where we see the sigma and the phi angles. And once our model is ready, then we go for the active site prediction, like active site prediction, that where the docking needs to be done, where we want to dock our molecule, because that is the site where the docking will actually take place, so that our molecule, whether it is an agonist or whether it is an antagonist, so it will either inhibit the activity, or it will enhance the activity of the protein. And I think
Yes, and for homology modeling, just a minute, sir. For homology modeling, you can use modeler, Swiss model. There are different tools available which can help you for homology modeling of novel protein.
yes, definitely. Now, I would like to make one thing very much clear, that when we yesterday we did the session in the introduction, like how we will be doing these sessions, and how we will be sending the feedback link for the certificate, and who is admitted into the session. But then, there are people who are saying that we have internet issues, and we could not attend. However, I could see as of now that there were 50 more than 50 participants who attended, but right now only 23 participants are left, including me, so, and including Miss Sharvari, and the company, the host. So there are 20 participants only left, who, as of now. So what, and people are, when we have sent the message for the feedback link for the certificate, then people are asking, people are still sending requests to join. However, that is not the way we do it. We do not sell the certificates, we sell knowledge, and knowledge right now, it is free. And that is the reason why we have put it on this session live on YouTube, so that you can at least gain from it. Now, if you want the certificate, there is a process for that. The process for that is that you need to go to the website, you need to fill out the registration form, and then you need to join the WhatsApp group. So all the information regarding the Zoom meeting link and everything that will be sent in that particular group itself. At least 3 emails were sent to all the participants who have joined this Zoom session. And in that, it is clearly mentioned that we will not allow any participant to join if they are late for more than 10 minutes, which means that the session started at 8 p.m. and we would not allow any participant to join after 8:10 p.m. But still we allowed people to join even after 8:30 p.m., even at 8:45 p.m. But then if you think that this is for the YouTube, obviously I'm just making it clear, this is for the YouTube, that if you are joining late, then unfortunately you cannot. When we have mentioned, there are certain rules and regulations by which we have to abide. So unfortunately we cannot go ahead and continue like this. We can only the feedback link is already been shared here into the Zoom, and the certificate will be sent tomorrow. Now if you still want to attend the same bioinformatics sessions who have missed it out, then we will launch it again in the first week of November. The process would be the same. You go to the website, you register for it, fill out the form, and then join the WhatsApp group. In the WhatsApp group, we will send you the Zoom meeting link, Zoom meeting ID, the password, and then you can join on time. So if you have missed it now, then and you want the certificate, then you can join the next session, or else the entire session of more than 3 hours will be available on our YouTube channel, so you can go ahead. Now one more question I have got is from Mr. Deetam Chakrabarti. Is model validation technique valid for unknown small peptides too? Sharvari, would you?
Model valid, yes. Yes,
Uh,
sir. If you want to proceed, you can, sir. Should
okay.
I answer?
Okay, so people who have not filled the feedback form, kindly fill the feedback form with the name and the details, and your certificates will be sent tomorrow by email. So thank you very much, and keep learning from BDG Life Sciences. I will ask my team to just put what I should say, the link of the upcoming session. So in the chat box, now you would have got the link of the website. So from here, you can register for all our upcoming hands-on technical workshops. So thank you very much, and keep learning from BDG Life Sciences. Stay home, stay safe, maintain social distancing, and most importantly, take good care of yourself as well as your loved ones. Thank you very much. Bye bye for now.